Now let's briefly talk about some of the equipment you'll be using in the lab. I would like to point out here that the photographs here and description is about generic equipment. The specific equipment that you will be using may vary from lab to lab. So again, please ask your instructor if you have any questions regarding the piece of equipment that you would be using. First of all, I would like to talk about laminar flow hood. This piece of equipment basically allows workers to manipulate their samples in an area where they won't have to worry about contamination. This is a two-dimensional photograph, but I would like to describe it so that you get the three-dimensional picture of it. Think of it as a big table, and this is the working area of this table. This table has barriers on three sides. So here's the first side, the second side, and of course one in the back. So there's only one side where there is no barrier because of course this is the place where a person will be sitting and working in this laminar flow hood. Laminar flow hoods basically work by jetting streams of air towards you or towards the room. There's a motor here which basically sucks in air and it basically filters that air so there are no particulate material in the air, and the jet stream basically are present at different locations also in the back of this equipment. So the back wall has little holes through which the air will be jetting out, so no particle can move from outside to inside this laminar flow hood. Of course, you want to keep it sterile. You have to first of all sterilize this laminar flow hood. We'll talk about sterilization later on. But anyways, once the laminar flow hood has been sterilized, you can basically open your samples, your cell cultures, for example, without having to worry about getting them contaminated. Because as we know, air contains spores of many different pathogens and different microorganisms that if those spores or those microorganisms enter your container, which has your sample cells, it can, of course, damage your sample. So let's talk about the next equipment, the incubator. Of course, all cells have different requirements. Different cells require different temperatures, and also they have other requirements. Human cells, mammalian cells require 37 degrees centigrade. Insect cells, for example, require 27 degrees centigrade. Incubator maintains optimal temperature and humidity for cell survival and proliferation. Incubator has two doors. The outer door is thicker and it insulates the internal environment, so temperature could be maintained. The second door is a glass door. Also present inside the incubator is a little dish which is full of water, a distilled water, which keeps the internal space of the incubator moist. So also different incubators may also have carbon dioxide function in them. They can regulate the concentration of carbon dioxide gas inside the incubator. Carbon dioxide gas is important because we can use carbon dioxide to maintain the pH of the media and the pH in which cells are growing. No cell biology lab will be complete without a microscope. We look at the cells and we need a microscope to look at the cells. Here I have pointed out a few important parts of the microscope. Here, for example, are the objectives. This part of the microscope is basically close to the sample, which is placed on the stage. The stage you can see here, this black part, and here is a dish which is transparent. Inner circle is a transparent. You will place your dish here, and right under the dish there will be objectives, and objectives have different magnification powers. You will select the appropriate objective. Objectives, the light is shown at the top, from the top on the sample. It enters the objective. Here there are mirrors which will reflect the light, and light will come out through the eyepiece. Here is the observer or the person who is looking at the cells, so they'll be able to see the magnified image of the cells. The light basically comes from the lighthouse. This housing has a bulb in it, and when microscope is turned on, the bulb will illuminate and provide light. The light intensity can be varied by placing different filters here. Excessive light can be toxic to the cells. So we can put different types of filters that will make the light intensity lower so we can observe our samples for a longer period of time. Diaphragm basically also manipulates how much light reaches the sample. Phase slider, if you are using phase optics on your microscope, basically allows you to see the cell with more contrast, and you can judge different structures in the cell. You can see them much better. Here's the condenser. This condenser basically focuses light on to the sample. You may have a camera port on your microscope which is used to take photographs of the sample. You need to focus on the sample. You need focusing wheels. There are two types of them. The inner one is the coarse focus, and the outer one is the fine focus. So you can look at your cells clearly. So this is what I wanted to talk about, basically say about the microscope. Let's look at the next piece of equipment, centrifuge. Centrifuge basically rotates the sample around a fixed axis, simulating high gravity, causing denser substances to sink to the bottom of the sample tube. This is fairly important. For example, if you want to wash the cells, you have a cell suspension, and you can pellet the cells by putting them in the centrifuge for a few minutes. And once you take out the sample, all the cells will be at the bottom of the tube. That has been achieved. So here's the palette of the cells, and here's the media that you want to change. You can easily remove this media and add new type of media or whatever materials you want to add to your samples. So this is an important piece of equipment. Let's now talk about water bath. In your lab, you will see this large container, basically made up of steel. This basically has sort of a similar function to the incubator. It maintains fixed temperature. However, it's an open space. Generally, you don't put cells in a water bath. This container is full of water. Basically, it is used to thaw samples. So if you have something frozen sitting in your freezer, you will place it in the water bath, and it will thaw it more quickly than if you left it out at the room temperature. Also, different enzymes we have talked about, they have a specific temperature requirement. And if we put our sample, for example, we need to digest a sample, we will add the enzyme and put it in a water bath. Please make sure that the temperature indicated on the water bath is the correct temperature you want your samples to be exposed to. You will see other pieces of equipment also in your lab, maybe different versions or additional equipment. Please ask your lab instructor to help you figure out whatever equipment that you need to use.